So the first talk on how phonological knowledge shapes early word learning in typical or atypical development and eye tracking study, and by Sonia Frota, Ivana Pejovic, Katia Severino, Marina Vigário. I can see the speaker is Sonia Frota. Sonia, over to you. Would you like me to play a sound when you have only 2 minutes left in your presentation time?
Oh, well, it's up to you. I'm timing myself, but it's up to you. Okay.
Okay, then let's do that. Okay.
Okay, thank you very much. Are you seeing my PowerPoint?
Yes.
Yes. And is it, oops.
Very clear.
Yeah. Okay. So, thank you again. And I'll be talking about early word learning. Developing a lexicon requires associating sound sequences to meaning. It is well known that infants demonstrate a perceptual sensitivity to speech sounds that attunes to the native language during the first year of life. However, how does the knowledge about the sounds interact with the knowledge about the words? This is a challenging research question that this study addresses. So I'm sorry, I could see that my PowerPoint was not moving. Is it moving now?
It is moving.
Okay, thank you very much. So, previous work has shown that early word learning is sensitive to native phonotatic patterns, with high-frequency sequences being learned earlier. Word learning is also possible with non-native phonological sequences, but not with phonologically illegal sound sequences. Moreover, similar sounding word pairs are especially challenging for the early learner. However, studies addressing legal or illegal phonotactics, to the best of our knowledge, did not use similar sounding pairs. There are good reasons for studying early word learning abilities. Word learning is critical for language acquisition, and the beginnings of sound meaning associations not only correlate with later language outcomes, but may signal risk for language impairments. The present study examines whether and how relative phonotatic frequency, meaning high-low probability sound sequences, and phonological grammar, meaning illegal sound sequences, impact early word learning in both typically developing and at-risk toddlers, and using similar sounding pairs in an eye-gaze task. The participants were all raised in monolingual European Portuguese homes and had no hearing or visual problems. We have two groups of participants. The typically developing group, born full term and with no familiar risk for language impairments, included 31 toddlers. The at-risk group, born premature or with familiar risk for autism or language disorder or other risk factors, included 29 toddlers. In this talk, I will mark the typically developing group, always in green, TD, and the at-risk group, AR, always in orange. We focused on infants over 18 months because previous studies have shown that they are able to associate objects with similar sounding words in laboratory tasks. The participants' eye movements were recorded while watching videos in a word learning task. For the test items, we used the visual stimuli from Gonzalez Gomez et al. 2013 that consists of 8 pairs of unfamiliar objects. We also used 4 pictures of familiar objects as control items. The visual stimuli were paired with speech stimuli produced by a female native speaker in child-directed speech. These included CV-CV disyllabic sequences embedded in natural sentences, and these sequences have high-frequency onset consonants and low-frequency onset consonants to manipulate phonotatic frequency, as well as an illegal prosodic word onset to test for phonological grammar effect. The vowels were always the same across conditions. Thus, test items were pseudo-words, in the case of high-frequency sound sequences like Sato, and in the case of low-frequency sound sequences like Raju, and were non-words in the case of illegal sound sequences like Nyago. Control items were familiar words to the children like bola, ball. The experiment included 8 blocks. Each block consisted of a learning phase, a test phase, and a control trial. There are 3 kinds of learning conditions: learning a high-frequency sequence, learning a low-frequency sequence, and learning an illegal sequence. In the learning phase, children are presented with one object that is labeled 6 times. This is going to be the familiar sound object pair. They are also presented with one object that remains unlabeled. In the test phase, children are presented with the familiar sound object pair and also with the another object that was unlabeled in the learning phase and that gets now an unfamiliar label. And finally, we have the control trial, where children are presented with one familiar word and respective picture. Let me play you an example of a block.
Olá! Vamos dar um nome a este boneco.
So we have a doll speaking.
Este é o Sato. Olha para o Sato. Vamos brincar com o Sato. Sato! Olha para ele.
This is the labeled object.
Sato é bonito. Eu tenho outro boneco. Também podes brincar com ele.
The unlabeled one.
Eu gosto muito do Sato. Olha! Onde está o Nhago?
The test phase.
Olha! Olha para o Sato. Olha!
And the control.
Olha para a sopa.
Okay. So the experiment has 8 blocks with side of target image and order of test trials counterbalanced within subjects. Target image sound association was counterbalanced between subjects and order of blocks was pseudo-randomized. We use an eye tracker to record eye movements. For data analysis, we measured net dwell time for each of the 2 dynamic areas of interest, target and distractor. And by target, we mean the picture that corresponds to what the infant heard in the trial. We computed proportion of looks at the target in 2 phases of the trial, the pre-naming phase, that is the part of the trial before the speech stimuli, and in the post-naming phase, that is the part of the trial from the onset of the speech stimuli until the end of the trial. Data trimming was similar to Gonzalez-Gomez et al. 2013, and we used linear mixed models for the analysis. If an overall learning effect is found, regardless of learning condition, this would indicate that learning is not guided by phonological constraints. If we find a learning effect modulated by type of sound sequence, in particular, distinguishing illegal versus the other types of sequences, this would indicate a phonological grammar effect. If we find a learning effect modulated by type of sound sequence, but in this case, distinguishing the high frequency sequences from the others, this would in turn indicate a phonotatic frequency effect. And finally, if there is no learning across conditions, then this might suggest that similar sounding pairs are too challenging for the children. So now the results. We first looked at the control trials and examined the proportion of looking time to the target in function of the trial phase, pre-naming, post-naming, and group. The linear mixed model analysis showed that both groups increased looking time at the target in the post-naming phase relative to the pre-naming phase. There were no differences between groups and no interaction. Now, for the test phase, the data of the post-naming phase in the test trials was explored to look for effects of the learning condition. So we want to know whether there are differences between the high frequency, low frequency, and illegal learning conditions. For the typically developing children, we found an effect of familiarity, and crucially, we found an interaction between familiarity and learning condition. Specifically, we found that the difference between familiar and unfamiliar sound object pairs was found only in the high frequency learning condition. For the at-risk group, no significant differences were found. Well, what does this mean? Our results have shown that for the typically developing group, learning is modulated by type of sound sequence, distinguishing the high frequency sequences from the others and thus demonstrating a phonotatic frequency effect. Whereas for the at-risk children, the results show no learning overall. The finding that word learning depends on the learning condition, with high frequency sound patterns facilitating learning and low frequency patterns behaving similar to illegal sequences, suggests that phonotatic frequency is guiding toddlers' early knowledge of the phonological grammar of words. The absence of a similar effect in the at-risk group might suggest either phonological delay or a different developmental path. Well, the toddlers' results that we have found differ from findings for EP, European Portuguese adults, in an EEG study that demonstrated a difference in the processing of high frequency, low frequency, and illegal sound sequences, showing the adult findings showed an early effect of phonological grammar that was followed by an effect of phonotatic frequency. This difference between the adult findings and the present findings suggests that early word learners follow initially a learning path guided by phonotatic frequency. In ongoing work, we are now examining the potential link between performance in the word learning task and vocabulary knowledge of the children. And in fact, we have some nice findings, and I may go into that if you are interested during the QA period. We are also investigating the same research questions that we addressed in the current study using EEG, as although eye gaze paradigms have been able to reveal early word learning abilities for similar sounding word pairs, it could well be the case that an EEG paradigm might be even more sensitive. But we do not have the results yet. And finally, I would like to thank all the children and their families that participate in the study, as well as our partners from Dona Estefânia Hospital in Lisbon and the Neurodevelopmental Center, Diferenças, that helped with recruiting the children. And thank you very much for listening.